Hi everyone! Today we will give you a short introduction to Illumina's method of next-generation sequencing. Next-generation sequencing, or NGS, is a method to sequence thousands to millions of DNA molecules simultaneously. Illumina is a biotechnology company that plays a large role in next-generation sequencing. To understand more what Illumina presents, this lecture will focus on the method of NGS, the controls that are being used, and what the diagnostic applications are.
Illumina is using the following 4 steps in their method: sample preparation, cluster generation, sequencing, and data analysis. These steps will be explained shortly. The first step is sample preparation. During sample preparation, a DNA sample is cut in several pieces, called fragmentation. There are a lot of different methods for fragmentation, but they will be not discussed in this lecture. Adapters are added to the ends of the DNA fragments. Adapters are single-strand or double-stranded oligonucleotides that can be ligated to the ends of the DNA fragments. These adapter sequences have an important role in Illumina sequencing because they hold barcoding sequences, also called tags, forward and reverse primers, and important binding sequences to attach the fragment to the flow cell and allowing bridge amplification. Step 2 is cluster generation. During cluster generation, samples are loaded on a flow cell. A flow cell is a glass slide with lanes. 1 billion of nanowells are present in these lanes. The wells of the flow cell are coated with oligos, primers. They are complementary to the adapters on the DNA fragments. The DNA strands are able to bind on the surface of the flow cell. DNA polymerase makes a complementary strand of DNA. The double-stranded DNA is denatured, and the original DNA strand is washed away. This amplified DNA strand bends over and hybridizes to the oligo that's complementary to the adapter on the other side of the DNA strand. DNA polymerase creates a double-stranded bridge structure, and this bridge is denatured. The result is 2 single-strand copies of the original DNA strand. This process is repeated several times. Reverse strands are washed away, and the 3' end are blocked to prevent unwanted priming. Several dense clusters are generated in each channel of the flow cell.
After cluster generation, lots of clusters on the flow cell are sequenced at the same time. With NGS, forward and reverse strands can be read, starting with the forward strand. Fluorescent nucleotides compete with each other to bind to the DNA, and while binding, a fluorescent light signal is emitted. The wavelength and signal intensity determines the base. After reading the whole sequence, the product with reads is washed away. To read the reverse strand, the oligo of the forward strand is binded to the flow cell, which again results in bridge amplification. After bridge amplification, the forward strand is washed away, leaving the reverse strand, which is read in the same way as the forward strand. This results in millions of reads containing all the fragments that were bound on the flow cell. Data analysis starts by organizing the reads based on the individual tags. Then, reads with the same length are clustered together. Forward and reverse reads are paired. At last, the data can be transferred, stored, further analyzed and shared. Aside from the basics to prevent contamination in the samples, such as eliminating air particles or contaminated lab equipment, it is also important to add controls. The following controls must be added during sample preparation: a blank sample, which does not contain the template, a positive control in the form of a known composition, and it is also critical to control for PCR inhibition and template occurrence, ideally by adding reference DNA to samples before extraction.
An example is the use of next-generation sequencing at the Netherlands Cancer Institute, where they use NGS for finding mutations, such as BRCA1 and 2 for breast cancer. The bulk of experiments are performed using validated protocols and kits. Also, a lot of articles are published thanks to the NGS technology. This was a short introduction to Illumina next-generation sequencing.